Hello students, we are back with yet another interesting topic, that is DNA replication. So before starting DNA replication, we need to know a little bit about DNA. DNA is a molecule with two strands wrapped around each other in a form of a double helix. Each strand has got a sequence of nitrogenous bases represented by A, G, C and T. If one strand has got G in it, the other strand will have C. Similarly, if one strand has got A in it, the other strand will have T. Each strand of DNA has got two ends, the 5' end and the 3' end, which runs antiparallel and opposite to one another. Now, DNA replication starts from a specific point which is known as the origin of replication or the ORI. The first step of DNA replication is separation of the two DNA strands with the help of the enzyme known as helicase. Now, the separated strands of DNA will serve as the template which will further provide information for formation of new strand, that is the complementary strand. Now, the initiation of DNA replication is done with the help of the enzyme primase, which adds a small stretch of RNA or D-primer opposite to the DNA template strand. The primer is attached to another enzyme known as DNA polymerase, which further elongates the new chain, that is the complementary strand, by adding the complementary bases opposite to the template strand. This results into formation of two types of strands. One is leading strand, the other is lagging strand. Leading strand is named so because the DNA polymerase continuously keeps on adding the nitrogenous bases, whereas the opposite strand, that is the lagging strand, is named so because small stretches of DNA is being added with a regular interval of time, within a regular interval of time. The small stretch of DNA is now known as Okazaki fragments. Now, as I've already told you, the opposite strand, that is the lagging strand, is named so because small stretches of DNA sequence is being added. Those sequences are known as Okazaki fragments. Now, as the name suggests, these are fragments, these are not continuous. These fragments needs to be attached with one another with the help of a glue. The glue in genetics is nothing but DNA ligase, which will join the consecutive adjacent Okazaki fragments to form a continuous stretch, completing the process of DNA replication. Now, at the start of the process, we had two strands of DNA, but now at the end, we have four strands of DNA. This is how a DNA content is being doubled, and this entire process is known as DNA replication. Wasn't it easy and interesting? For more such videos, subscribe to our channel. Thank you.